Hi, everybody. It gives me a lot of pleasure to introduce this topic. What if your response is a curve? I think this applies to a lot of biological systems. Certainly, we have encountered it many times at Synthace, where you're looking at a growth condition or a binding condition, and ideally, you'd want to be able to put the whole thing into your response, and usually you have to pick some kind of feature of that curve to try and analyze in normal sort of DOE methodologies, or so we thought. Because Phil is here to obviously tell us about how we can do things better, which I'm genuinely very interested in. So as Khalid said, please take the opportunity to ask questions as we go. We'll address them at the end, but ask them as they occur to you in the Q&A box. Now I'm excited to introduce our presenter for today's webinar. It's Dr. Phil Kay, who's the Learning and Development Manager at JUMP. So Phil is the Learning Manager for JUMP, which is a division of SAS. His job is to understand scientific and engineering challenges and provide guidance on data analytics solutions for industrial organizations all around the world. The experience that enables him to do this is on the basis of being a development scientist for numerous processes for the manufacture of colorants for digital printing at Fujifilm. Phil has a master's degree in applied statistics with a dissertation on design of experiments specifically, and that's all based on a master's and PhD in chemistry. So we're in really good hands. Phil, thank you very much for joining us, and without further ado, I'll hand over to you.
Thanks very much, Marcus. It's a real pleasure. I'm a big fan of these DOE Masterclasses. So I'd recommend anyone to go and see the previous ones if you haven't done so already. So I believe we're going to start with a polling question. So I was interested to know to what extent design of experiments is used at your organization. So you've got 4 options there. It's either your default way of doing experiments, you use it on special occasions, you've used it but it didn't work for you for some reason, or you've never tried it. So please let us know through the poll here. So I'm hoping that there's going to be some of you there have got quite a bit of experience in using design of experiments. If you are new to design of experiments, then welcome, and I hope you're going to find this useful. I'm just waiting for the results. Great. So, okay, so quite a lot of you, 25%, is saying you use it as a default method for experimenting, which is wonderful. That's kind of been my my mission most recently in my career, really, is to get people using design of experiments as their default way of experimenting. 60% say you're using it on special occasions, so that's interesting. And the 16% that have never tried it, and maybe the reason you haven't tried it is because you couldn't see how it would work in your case. Maybe you've got some special case that you didn't think that design of experiments was going to be able to help you with. Well, I'm going to be talking about some of the special cases where in the past it was hard to apply design of experiments, and show you how with different methods that are cutting edge, you can actually tackle these problems now. Right, so let me, hoping I can share my screen. And hopefully you're able to see my screen now.
Yep, all good.
All good, great. So yes, so the question is, how can you apply design of experiments if your response is a curve? I wanted to start with some personal experience from my time when I was working as a development scientist in the lab. And the kind of experiments I was doing were quite dirty, kind of crude things involving milling of pigment dispersions. So this diagram here shows us the idea of a milling, a bead mill. So what we have is we have a dispersion of the pigment, so it's dispersed in water with various other additives, and you pump it through this mill, and in the milling chamber there's beads, these hard beads, small beads that are being rotated at 4,000 RPM. They're smashing against each other, they're smashing into the pigment particles, and they're grinding up the pigment particles, and you continually circulate the pigment dispersion through this mill. Now, what you want is to reduce the particle size as quickly as possible into the target range. So it's not, may sound crude, but it's not simple. It's quite a hard problem to work out the best settings for that. So we use design of experiments. This is a case where you've got multiple factors. You've got things like the volume of milling chamber that you fill with beads. You've got the strength of the pigment dispersion. You've got how quickly you flow the material through, and you've got a temperature. You can hold the vessel at a certain temperature, which will affect the viscosity and therefore affect the milling performance. So we're trying to understand how varying these factors will vary our ability to get the target size, the particle size, into the target specification. So what we do is periodically throughout this process, and it takes a few hours, we take samples, we take the sample off to the analytical lab, and we measure the particle size, various different measures of particle size. So what we get for each run of our experiment is we get one of these, what we call a milling profile. We get this curve. We can see how the particle size varies as a function of time. And what I was really interested in is how the factors, the DOE factors, affect the shape of this curve. But I didn't know how to do that. I didn't know if there were any tools that would enable me to do that. I knew about design of experiments. I knew how to model design of experiments when you've got a simple response that's just a single number. But try as hard as I couldn't find a way to actually answer the scientific question that I had, which was how do the factors affect the shape of this curve? And this kind of curve data is everywhere in science and biology. Spectral data, so where you're looking at absorbance intensity as a function of wavelength, wave number. Chromatography, where you've got some kind of measure of intensity or other against the retention time. And then in biological processes like fermentation, you're often looking at how things are growing versus time, and you'll take samples, you'll take measurements throughout the fermentation process. You know, things like OD as a measure of cell growth. So these curves are generated in lots of places. A lot of the time, the thing that we care about is some kind of curve or functional data, as we call it. Now, I know at Synthace, you've got your own lab for trying out new things, trying out new ideas. This is actually, it's a long time since I've worked properly in the lab, but this is an experiment that I've done it in my own personal research kitchen, along with my lab technician helper. And we were looking at an experiment to see what factors affect cress growth. How do different factors, growing conditions, affect the growth of cress? And it's a fun little experiment. The idea was to try and come up with an experiment that's easy for people to do at home, so they can try out design of experiments. And you know, it's cheap, simple, and easy enough for children to get involved. So we've set up an experiment. We have 12 runs or 12 pots. So this is my lab technician helper there that you can see. The great thing about this as a home experiment is that you can actually reliably, even if you've got a really rubbish lab technician like I've got, you can reliably get measures of the response, which is actually what we do is we measure the tallest plant in each pot, the tallest cress plant. And it's nice, the way we've done it, you can use some old egg boxes. So instead of 96 well plates, you've got these egg boxes, which are basically 6 well plates. And I'll give you a reminder of this. And this is actually part of a summer DOE challenge that we're doing at Jump. You've still got a little bit of time to register for this, and I'll talk about the competition we're doing later. So this is the version of the experiment that we did. 3 factors, 12 runs. So the factors were the substrate, what I'm calling the substrate, so either soil or cotton wool. 2 different light conditions, basically growing it in the light or in the dark underneath a box. We've got the cress type as well, so we bought seeds for 2 different types of cress, plain or curled. And with 2 different choices for each of 3 factors, there are 8 possible treatments. So the full factorial for that is 8 treatments. We're actually repeating 4 of those. So we're repeating the half factorial there. So 12 runs in total, so it fits nicely into 2 egg boxes. Why is this relevant to what we're talking about today? Well, what we did was we measured the response, the maximum height, the tallest plant in each pot. We measured that on days 0, 1, 2, 3, 5, and 7. So this is functional data. This is data that forms a curve. It forms a growth curve. So for each run, each of our 12 runs, we've got this growth curve. You can see how the plants have grown in each of those different conditions, those different combinations of factor settings. So what we'd really like to do is to understand how the factors affect the shape of those curves. That's fundamentally what we're interested in. So it's going back to the same challenge I had with the milling experiment. How do we manage that? How are we able to do that? Well, I'm going to talk about 3 options for doing this. I'm going to talk about what you can do just with standard design of experiments analysis. Many people have been successful in modeling these kind of things using standard DOE analysis. I'll talk about the advantages and disadvantages of that and of the other methods. I'll talk about an idea called curve DOE analysis, or what is sometimes called hybrid modeling. And finally, functional DOE analysis. So let's start just with standard DOE analysis. So how would we model this just using our standard DOE modeling tools? Well, we would model the factors against some responses, right? And what we can do here is we can just say the height on day 3, the height on day 5, the height on day 7 are 3 separate responses, and we just model against those responses. We see how the factors affect each of those responses individually. In Jump, we can combine that into the same visual here. So this is a prediction profiler. So this is how we model, how we visualize these models in Jump. So you'll see more of this prediction profiler. So you see the factors on the horizontal x-axis, and we've got our 3 responses on the y-axis. And so we can visually see how these factors affect the responses. You can see that light condition is the most important factor. We can make some prediction about what the maximum height will be on days 3, 5, and 7. You can see that cress type is the least important, maybe isn't really a significant factor here in governing the height on day 3, 5, and 7. And actually, if you're doing some statistics, if you want to keep your statistics colleagues happy, then you should do something called false discovery rate statistics here. So that's what the FDR p-value means there. It's nothing to do with Franklin Delano Roosevelt. It's a way of making sure that you're not cheating the statistics when you're modeling more than one response. So that's good. I mean, that is an insight. There's nothing wrong with that. What it does rely on is having aligned time points. So if for some reason we weren't able to measure the height on the same days for all the pots, if we don't have aligned sampling points, which you may not have in other situations, then we wouldn't really be able to do this. It relies on having the same response for each of the factors, for each of the runs, rather. And also, it's not really answering my fundamental question here, which was, how do the factors affect the growth? You know, it's telling me how the factors affect the height on days 3, 5, and 7, but it's not really getting to the heart of my scientific question. So there's the advantage here is that it's simple, standard modeling, but the disadvantage, again, we need aligned time points, which was trivial in this case. That was easy enough to do, but won't always be the case. And it's making it harder to interpret, make sense of what's really going on, and relate that to our real scientific objective. So let's look at curve DOE analysis, or what you might call hybrid modeling. The idea is here for each curve, so we've got our 12 curves for the 12 different runs, 12 different pots of Cress. We fit some kind of mechanistic model to that functional data. So I'm going to start by just fitting probably the simplest model we could fit, just a straight line fit. Now, it's not a good model for this data, but just to illustrate what we're doing here, let's start simple. And now for each of those runs, instead of having this curve, we can summarize that in terms of the intercept of that straight line fit and the slope. So how steep that slope is, which is a crude, it's not ideal, but it's a crude measure of how quickly the cress is growing in each of those pots, I suppose. The intercept doesn't really make sense in this case because it should just be 0 in all of these. So again, this isn't a great model for all that functional data, for that growth curve. But as an illustration, what that then enables us to do is we can take those model parameters as our responses. So instead of fitting our factors against the responses, the height at day 3, 5, and 7, we fit against those model parameters. So we look to see how the factors affect the slope of that model, the slope of that crude representation of the growth curve. And again, that gives us some useful insight. It's kind of in agreement with what we saw before, crest type being the least important and the light condition being the biggest factor. Now we can do the same thing, but with a greater degree of sophistication. So we could fit a more meaningful mechanistic curve model to that functional data. So this is a 4-parameter logistic curve, I think. Can't quite remember exactly what model. There's a variety of different models that we might fit to this kind of growth curve. And we've got more parameters now, and those parameters have got some more meaning to them, actually. We've got a growth rate parameter, the inflection point, and then the lower and the upper asymptotes of that curve. So we can take those now, and we can model the factors against those. So we've got different parameter estimates for each run, for each pot of cress, and we can use those as our responses. Model the factors against those as we would in a standard design of experiments model. What's nice about this is that you can then ultimately reconstitute that curve. So you can say, because we now have a model that tells us how the factors affect the parameters of that curve, we could actually reconstitute that curve. We can actually recreate that curve for any of our factor settings. So here you can see what I've done is I've got the prediction profiler there with that first blue box is actually that growth curve, that modeled growth curve. And you can see how changing from cotton to soil, from the upper to the lower profilers there actually changes the shape of that curve. In this case, you can see it's clearly changing the upper asymptote. So the maximum height we expect to be higher with soil as the substrate rather than cotton wool. So again, just to repeat, the idea being there that we model parameters from a fit of a mechanistic curve model to the functional data. So the big advantage there, as I think you've just seen visually, is we can model that curve as a whole. In many cases, if we've got a good enough model, a good enough mechanistic curve model to fit, we can get a more meaningful interpretation now and potentially a mechanistic understanding. We can see which factors are affecting the growth rate, which factors are affecting the upper limit, the upper asymptote. Now, some disadvantages there. It's not going to work in every case. We need to have a curve model that fits. And actually, if we just go back here, this model fit well for most of my runs, but not for run number 8. I think you can see there's just the fit there doesn't look very good, and maybe we needed a model that could actually model a kind of death phase, because it looks like the actual maximum height was decreasing later on for pot number 8, run number 8. So it does rely on having a curve model that fits to the data, that's a good model of whatever is generating, whatever process is actually generating that curve. It's a bit more time consuming. You can see that there's some other steps to that. You know, obviously I work for Jump. I'm going to tell you that Jump Pro has some nice tools to take a lot of the work out of that. It's actually a very straightforward process, and I'm sure there's other tools out there that can also help you to do these things. But you know, in some cases you might, depending on what software you're using, you might need to use separate software to fit the curve model and then model it against the DOE factors, and then working out how you reconstitute that into the actual curve at the end could be a challenge. There's a few steps to be taken there. Okay, number 3. So this is the final method that I'm going to talk to you about, which we call functional DOE analysis. And I should say this is the language that we use at Jump. It's not necessarily the language that's used more broadly. I'm not sure if there's necessarily a consensus on what the right term is to use for these different modeling techniques, because a lot of it's quite new. So what's different about this? Well, we're going to start by fitting a smoothing model to the functional data. It's not a mechanistic model, it's just an interpolation model. And you know, when I say smoothing model, you might imagine something that's a bit smoother than this. Actually, this is quite jagged kind of model, but it's basically taking our semi-continuous discrete data at different time points and converting that into a continuous function, continuous curve, or jaggedy line as it is in this case. What we then do, having done that, is use some clever analytics, some principal components analysis, in fact, functional principal components analysis. We don't have time to talk about how that works, what it does, but I can tell you, why we're doing that is what it enables us to do is to decompose the shape of those 12 curves in this case into the mean. So we've got an average curve. This is the typical. If we just average how the height is changing as a function of day across our 12 pots, this is what the average looks like. And then what we say is, well, there's obviously variation from that average. That can be captured, that can be explained, can be modeled in terms of different amounts of the characteristic curve components. So it finds these characteristic curve components. And what we're saying is that each one of these original 12 curves can be described as a combination of the mean plus or minus some amount of component 1, plus or minus some amount of component 2. And component 1 is the component that's describing the most variation. Then it'd be component 2, and then potentially 3, 4, however many you need. In this case, 2 components does a good job. In fact, it describes over 99% of the variation in these curves. So again, just to reiterate, we can describe our 12 individual curves now in terms of the mean curve, the mean growth curve, plus some amount of component 1 and some amount of component 2. So it looks something like this. So for each run, now we've got a weight or an amount of each of those curve components. And that's what's characterizing the variation. So if you look at these Cress pot number 4 and number 8, you can see they're similar growth curves. There's a little bit of difference at the end there, but they've both got a very similar weight for curve component 1. These are the ones that grew the most rapidly and grew to the highest ultimate height. And you can see that that makes sense. If you take the mean curve, you add component 1, if you add lots of component 1, that's going to give you quite a steep curve. In contrast, if we look at numbers 3, 6, and 12, they've all got around the same score there for curve component 1, and it's a low score. So if you took the mean and you subtract, it's actually a negative here, so you subtract component 1 from that mean curve, then that gives you curves 3, 6, and 12. Gives you the growth curve for those 3 pots of cress that all showed a similar kind of growth rate and a similar ultimate height. So we are able to describe this curve now in terms of the amounts of these curve components. So now those amounts of the curve components become our responses that we model against. And again, having done that, we can reconstitute what the curve would be for any factor settings. So potentially for factor settings that we haven't tried. In this experiment, we tested all possible combinations of the factor settings. In other experiments, you might have continuous factors, so you can actually make predictions about what the curve would look like for factor settings that you haven't actually tested. They're just between the range of a continuous factor like temperature that you've tested. So again, the amounts of each curve component and then model as the responses. And again, that enables us to really answer the question that we've got, the scientific question that we have. So yeah, how does each factor affect the shape of my growth curve? And I can see here, you can see the growth curve that we've modeled here, the predicted growth curve in the blue cell there. You can see how that changes as you go from light conditions in the top plot there to the darker conditions in the lower plot there. So you can actually directly see how changing factors affects the shape of your curve. And in this case, as I say, it's a growth curve. It's a fairly trivial experiment. But imagine how this applies to your examples where you're looking at something, whatever your curve might be. It might come from a biological assay. It might come from things that you're measuring over time versus time during a fermentation process. It might be spectroscopy data. The key thing here is we're really able to answer that question that I think most scientists have in this situation. How do the factors affect the shape of my curve? And then, in some cases, you've got an ideal curve. You've got what you might call a golden curve. And you can actually then see how you need to set the factors in order to achieve that perfect curve that you want to achieve. So in my milling example, with these tools now, if I could go back 10 years to when I was actually doing those experiments, I'd be able to properly model the data and answer the question I have, which was, how can I get to this perfect milling curve where it drops really rapidly and then it gets into the target particle size zone and remains within it forever? That would be my ideal golden curve. And that's probably not absolutely achievable, but I can now answer the question, what factor settings will get me as close as possible to that golden curve? How do I need to control the factors that we can vary in order to give me a consistent milling profile? Which of the factors are the most important in determining the shape of that curve? So I think this is, from a personal point of view, really powerful stuff, and certainly people that I speak to in the scientific community, science and engineering, they're finding all this extremely useful. So just to summarize that again, the functional DOE analysis, what we're modeling as the responses instead of just our traditional single number responses. What we've taken then is these characteristic curve components of our curve using functional principle component analysis, and then we use the weights of each characteristic component for each run as the response that we model against. And again, as with the curve or hybrid modeling analysis, this enables us to model that curve as a whole. An advantage with this one versus the curve DOE analysis is that it's flexible to any functional data form. So we don't have to have a specific mechanistic model that's going to fit to that curve. It's much more generalizable. Now that's an advantage. It can be a disadvantage. You know, it might be too flexible sometimes. You need to be careful that it's not going to lead to kind of spurious predictions. It's not suggesting, you know, impossible growth curves or you know, whatever kind of behaviors that you're modeling with this functional data. Also, you know, it's less, you don't get that direct interpretation of the meaning of the functional form as you do with the curve analysis. You know, you don't have a growth curve parameter, but it might, you know, tell you other things that you wouldn't otherwise capture. Again, you know, that's quite complex to do without the right software. So, you know, that's a potential disadvantage. Again, we've got good tools in Jump Pro that are set up for this, and we've got a very active research group that are looking at improving these tools all the time. So, you know, just to summarize, you know, what I've shown you there is what you can do when your response is a curve. So I've gone through the three options there. The standard analysis, simple, easy to use. The disadvantages being, you know, you need these aligned time points or whatever your x-axis is for your curve. The data points need to be aligned in order for that to be meaningful. And it's not really, in most cases, giving you a direct answer to your scientific question. The curve analysis, enabling you to model the curve as a whole with meaningful interpretation, as long as you've got a curve model that fits the data. And finally, this most flexible functional data, functional design of experiments analysis, which enables you to really model any functional data form. And with